Bismillahirrahmanirrahim, and welcome back to this next video. And this is another video in the series of videos on the bioinformatics. In my previous videos, I'll share the link in the description. I've told you that the bioinformatics it usually started with the sequencing of the proteins or analyzing the protein sequences, because at the start most of the scientists they were interested in determining the 3D structure of the protein. They identified that the proteins that is a macromolecule. It is made up of amino acids, and these amino acids are of 20 different types, and these 20 different kind of the amino acids they are used for the synthesis of the protein. I also told you that the when you talk about the sequencing of the proteins, you usually start from the N terminus of the protein and you move along towards the C terminus of the protein. Then I've told you that the amino acid sequence of a protein that makes the primary sequence of the proteins, and these amino acid, the sequences of the amino acid, they adapt to different 3D structures, and then those particular 3D structure are responsible for the function of a particular protein. Beside the proteins, the bioinformatics is also focusing on analyzing the DNA sequences. So in this particular video, I'm mainly focusing on what DNA is. So during the 1950s, like as I've told you, much of the scientists like Kendrew and Perutz, they were struggling to determine the first 3D structure of the protein. On the other hand, the other biologists, most famously the experiments of the Griffith experiments, they were very important. So the other biologists, they acquired a lot of indirect evidence using the genetic experiments, and they identified that the DNA. Now we know that the stuff that make our genes is the DNA. So they identified the DNA, just like the protein, is also a macromolecule, and it is made up of 4 constituents nucleotides, which are symbolized by the letters A, C, G, and T. So as the proteins, they are made up of the amino acids. So the DNA that is going to be made from the nucleotides. So what are nucleotides? When you look at the general structure of a nucleotide, there are 3 parts. One is a nitrogenous base, the other one is a pentose sugar, and the third part is a phosphate group. So this is the general structure of a nucleotide. So the 3D structure of the protein, as I've told you in my previous videos, that in 1951 the sequence of the amino acids that was determined, and then the Kendrew and the Perutz, they identified the 3D structure of the proteins. However, the molecular biologists had to wait until much later, like the 1970s, to be more precise before they could determine the sequence of the DNA molecule and get direct access to the sequences of the gene nucleotide. So the scientists, they were able to identify the sequence of the proteins, but they have to wait until 1970s before they can determine the sequences of the DNA molecule, because the techniques that were developed for the sequencing of the DNA molecule, they were there in around 1970s. So this was a revolution, because A. Sanger, he already won a Nobel Prize, but this sequencing of the DNA, it won him another Nobel Prize, because the small DNA alphabets, like as I've told you, it is made up of the 4 nucleotides as compared to the 20 amino acids. So these 4 nucleotides, they were much simpler and faster reading, and quickly lent itself to the complete automation. So instead of going for the automation of 20 different amino acids, it was much easy to go for the automation of the DNA sequences, which are made up of these 4 nucleotides. So currently, if you look at the worldwide rate of determining the DNA sequences, that is much faster than the rate of the protein sequences, because when you are going for the sequencing of the protein, you have to focus on these 20 amino acids. When you are going for the sequencing of the DNA, you have to focus on these 4 nucleotides. Now, in my previous videos, I've told you that we always read the sequences of the DNA from the N terminus towards the C terminus. So what about the DNA sequences, how we read them? So in my next video, I'll be focusing on how you are going to read the DNA sequences the right way.